the next talk, a little bit out of order, is Erica Herzog from Yale, where she is the director of the Translational Lung Research Program and director of the Interstitial Lung Disease Center of Excellence. And she's going to be talking today about immune and inflammatory cells in IPF or ILD, but she could probably talk about anything because she knows about all the cells, right?
That's right.
Yeah.
So I'd like to thank the organizers, probably none of whom are here. I'd like to thank everybody for staying. And without further ado, I'll move into my presentation. I assume the green is moving forward. Yes. So I do have some disclosures. I consult for Boehringer in the area of safety in clinical trials. And I have received grant funding from several entities, though none of these entities have influenced today's presentation. First thing I need to do today is I need to thank my son. My son needed to have urgent surgery last week, and he can't walk because his leg has to be immobilized. And I discussed with him that I needed to come here today, and should I stay home? And he said, no, pulmonary fibrosis sucks. You have to go. And my daughter, who's standing there next to him, said, don't worry, Mom, I'll help Dad. Everything will be fine. So I really wanted to thank both of them for stepping up today so I could be here. The next slide is going to be a tearjerker. I wanted to just remember Andy. 2 years ago, I moderated this session with Andy, and he will always be missed. Okay, so now moving into what I'm really supposed to be talking about, which is immune cells and pulmonary fibrosis. The immune system is designated into two main arms. I'm talking about professional immune cells. I'm not talking about non-professional cells like epithelial cells or fibroblasts. I'm talking about, I'm talking about bone marrow-derived cells, which are divided either into innate immunity or adaptive immunity. And so the innate immune response occurs immediately. It's a cell, like, for example, a macrophage that recognizes a pattern in, say, an invading microbe or a tissue factor that's been released that shouldn't be there. And the cell realizes something is wrong and enacts an immediate protective response that happens immediately and then goes away pretty quickly unless it's perpetuated for some reason. This is in contrast to the adaptive immune arm. This is more like lymphocytes and adaptive immunity. This is an antigen-presenting cell recognizes an invading, or recognizes an antigen, processes the antigen, activates a lymphocyte, which then undergoes DNA rearrangement, and then sort of becomes a permanent part of the immune system. Innate adaptive immunity takes several days to begin and to start to have a response, but once it's there, it lasts longer. And the adaptive immune system then typically will activate effector cells to do their job. So for example, here's a lymphocyte telling a macrophage to do something. So all of this has been studied in IPF to some extent, and I'm just going to talk about some representative adaptive immune concepts and innate immune concepts in the context of idiopathic pulmonary fibrosis. So starting with adaptive immunity, the reason why I'm talking about adaptive immunity first is because it's, as you'll see at the end, it's a little less, probably less important, or at least in our current concept. So I thought I'd start with that and then end with innate immunity, since that's really what I want you to remember from today's talk. So here's a schematic of the lung and what, for example, these are CD4 lymphocyte populations. So here's the alveolar space, here's a blood vessel, and here's the interstitium. And the classical paradigm of what CD4 T helper cells might be doing in pulmonary fibrosis is that Th1 cells might either be causing the injury here in the alveolar space, or they might be secreting, say, gamma interferon, which might actually be anti-fibrotic. Th2 cells, which are more allergic type of cells, they make IL-4, they make IL-13. Those are thought to be maybe more stimulating in terms of driving fibroblasts to proliferate and differentiate and to make a lot of collagen. Th17 cells, this is a little more in the autoimmune family, hasn't been looked at so much in terms of IPF. And Tregs, regulatory T cells, are sort of thought of as maybe keeping the rest of all this stuff in check. And so they're regulators that maybe will shut down these responses when they're too much. And so the idea is that the idea in IPF for a while was that regulatory T cells might actually somehow be involved in pathogenesis. So the concept for the IPF Treg story first came almost 10 years ago now. I cannot believe this is 10 years. I remember like it was yesterday when this came out. This is from the Katsianides group in Greece, demonstrating that relative to control patients, IPF patients had fewer Tregs in their, defined by certain intracellular and surface markers in their BAL and in their peripheral blood, and that these Tregs, their suppressor function was actually impaired, meaning that the cells were both reduced in function and reduced in number and also reduced in function. And so the idea was, oh, well, maybe this dearth of functional Tregs might be important. At the time, I was also starting to look at lymphocytes, and so we were looking at Treg heterogeneity, because just like in fibroblasts, there's not just one fibroblast in IPF. There's a lot of different fibroblasts, and just like epithelials do a lot of different things, Tregs might be doing a lot of different things. So we were identifying Treg populations, and we identified what we called a rogue Treg, meaning it didn't behave properly. So we found normal Tregs that were functionally normal, and then Tregs that were functionally abnormal, and we identified a cell surface marker on these Tregs. It was called semaphore and 7A, but that's not important. The concept is that there were normal cells and there were abnormal cells. And that this Treg population, the rogue, was highest in patients with reduced event-free survival. So when we took patients in the Yale cohort and we stratified them into stable versus progressive, we found the progressive patients had higher levels of these rogue Tregs. And we also found that they might be mechanistically related to pulmonary fibrosis, because when we injected these cells into mice, which I know we all said isn't really a great model, but I can't inject Tregs into people, so I need some type of non-human-based model here that's in vivo. And we found that when we injected these rogue Tregs into mice, they actually developed mild pulmonary fibrosis, suggesting that there was some type of mechanistic connection between these abnormal regulatory T cells and the fibrotic state in the lung. This is quantification of what we, okay, this is just quantification of the collagen that we see here. And so you can see it was statistically significant. So we also found, we looked at it the other way. We said, okay, well, you have abnormal Tregs, maybe you develop fibrosis. What happens if you take away the normal ones? So in another study, we used antibody-mediated method to remove Tregs, and we found that this caused both the increase in pro-inflammatory mediators as well as augmented lung remodeling and collagen accumulation. So for example, removing the Tregs via this method resulted in increased CD40 ligand, increased IL-1, increased TNF-alpha, and increased IL-12P70, all of which are pro-inflammatory, though maybe not particularly pro-fibrotic. We also found that when we depleted this was a TGF-beta overexpression model, we found that when we depleted the regulatory T cells, that there was more collagen, as evidenced here on this trichrome stain of blue. And this was quantifiable. So here we're comparing the TGF-beta overexpression model with and without the Treg block, and we found that there was an increase in collagen. So T cells in IPF, like people were at the time were thinking about it, and Naftali already talked about his 52 gene signature, his transcriptional profiling of patients with pulmonary fibrosis who went on to do worse. What he didn't really mention so much was that the dominant signal in this 52 gene signature is actually from is from CD4 T cells. And it looks like there's some type of abnormally activated, hyperstimulated, somewhat senescent T helper cell phenotype that, if you want to go out on a limb, you could say, well, maybe there are too few Tregs, and that's why this is happening. But so Naftali showed that the blood of patients with progressive IPF does show this abnormal phenotype. He showed it in a Science Translational Medicine paper probably 4 or 5 years ago now. And then just recently, he just showed that beautiful data with the 6 heat maps that were identical. So that does suggest that the T helper cell populations might be important. But when you think about what we've seen in people, it hasn't played out so well. So think about the Panther trial. That was treatment of IPF patients with lymphocyte modulating agents like low-dose steroids and azathioprine. Patients actually did worse. What about treatment with the etanercept or TNF-alpha inhibition? That was 10 years ago. We already knew in 2008 that TNF-alpha inhibition, that's a Th1 secretory profile. We already knew that that didn't work. And just recently, this was really sad. I think we all thought that IL-13 inhibition was going to be efficacious. IL-13 is a prototypical Th2 cytokine along with IL-4. Blockade of IL-13, Kevin Flaherty ran this trial, was not effective in the treatment of patients with IPF. So I think the story on lymphocytes and adaptive immune responses in IPF is going to be very complicated. And while all these lymphocyte populations might be important when they work together, I think maybe suppressing them isn't going, at least currently, hasn't been shown to be beneficial, and we should potentially, at least in the short term, be looking elsewhere or be performing sort of more high throughput studies in humans to understand exactly what's going on in the lung. So let's switch gears now and talk about innate immunity. Innate immunity, I mentioned, is this immediate response. So that's, you have cells that are floating around with what are called pattern recognition receptors that are just waiting to find a pattern that they will recognize and they will respond to. And so these cells, I'm going to be talking about macrophages, but there's actually, monocytes, mast cells, dendritic cells, lots of different innate immune cells. But the story of macrophages in IPF actually dates back to the early 80s, and somebody already mentioned Peter Bitterman talking about inflammation in IPF as early as, I guess, the early 80s. He was the first person to actually show that macrophages and fibroblasts had interactions, and he's now moved on to be a fibroblast biologist, but I don't think that we can understate Peter Bitterman's contributions to the IPF field, and I wish he were here to contribute to this meeting. So, we'll be talking about studies of macrophages and more current studies of macrophages, but again, the macrophage story does date back decades to the early 80s. So thinking about, okay, you have an innate immune cell, and an innate immune cell is driven by activation of pattern recognition receptors. These pattern recognition receptors respond to patterns. They respond to either pathogen-associated molecular patterns, which are repeating patterns that you find in bacteria, like LPS, or certain sugars that you see in gram-positive bacteria, or CPG, unmethylated CPG islands that you see in bacterial DNA. Or they respond to danger-associated molecular patterns, which are either intracellular substances that are released after necrosis, or like cleaved extracellular matrix that shows that something wrong has happened and that your matrix is now being digested. So our innate immune ligands present in IPF, this is actually very recent data. So IMRI has shown via, I don't know what this is supposed to show, but this is IMRI's paper from earlier this summer, and these blue triangles show in IPF BAL that there are certain bacterial components that appear to be able to function as PAMPs. And this is all related back to the CPG DNA that I mentioned. We looked at it from a different standpoint. So, the microbiome is a really big deal in IPF right now, but I'm still very interested in the relationship between injury and repair. And so we looked at it as from the danger-associated molecular pattern standpoint and looking at DNA derived from, say, mitochondria. Mitochondria have unmethylated CPG islands in their DNA, and this gets released in the setting of injury. We actually showed very recently that, that the blood of patients with rapidly progressive IPF contains increased concentrations of mitochondrial DNA, which functions as a damp. And so this was found in both the Pittsburgh cohort and in the Yale cohort. And the red line here is a Kaplan-Meier curve showing that patients with higher concentrations tended to do worse. So yes, the innate immune ligands are there. So now potentially are the innate immune cells doing something funky? And the answer is yes. So thinking about what might macrophages or innate immune cells be doing, At the time we wrote this review 7 years ago, the concept of classical versus alternatively activated macrophages was very robust. And the idea at the time was that in the setting of tissue injury, you developed a classically activated macrophage phenotype, which is macrophages come in, they want to kill something. After they've killed whatever they're there to kill, then the tissue milieu changes, and the macrophages switch to an alternatively activated phenotype. I think a lot of work has shown that it's not dichotomous, and so it's not like you just have a classical and an alternatively activated. But you might have macrophages that want to promote healing or promote fibrosis, and so I'll call them more profibrotic, but just for the purposes of this talk, thinking about it as what the alternatively activated macrophages do, which is to exert an anti-inflammatory repair response and to participate in the accumulation of activated effector fibroblasts, I believe that it's helpful. So we started looking at macrophages and monocytes, which are circulating precursors of macrophages, in the IPF disease state. And what we found was that that IPF monocytes and macrophages at baseline are very hypermigratory, so they're in a hurry to get somewhere. And when you stimulate them, they go even faster. So whether this means they transit from the circulation into the lung more quickly and cause disease, maybe. Maybe they move more quickly within the lung, but there's clearly some type of migratory phenotype going on. They're also abnormally activated. So when you look for things like the mannose receptor or other types of mediators, they do express more. It's quantifiable. And so this is IPF lung, but also in the IPF circulation, when we compared circulating monocytes from patients with IPF to age-matched elderly or advanced age, very healthy controls, we found that there was a significant increase in monocytes that were expressing markers of alternative activation. These cells also displayed profibrotic functions. So, for example, when we separated the IPF cohort into stable versus progressive, meaning patients who were stable at a year versus patients who actually went on to have a clinically significant event, we found that the concentrations of these CD206 positive or mannose receptor high monocytes were elevated in the blood. We then went on to query whether or not there was a functional ramification of this. So we created a culture system where we took lung tissue, we decellularized it to make a 3D matrix. So this is, for example, human lung. Now we have a decellularized sort of 3D matrix. We put fibroblasts in, and then we put CD206 macrophages or CD206 low macrophages in the bottom, with the question being, do the macrophages interact differently with the fibroblasts depending on what their phenotype is? And what we found was relative to fibroblasts grown with CD206 low macrophages, that the addition of CD206 high macrophages to the culture system resulted in accumulation of fibroblasts that was quantifiable. So this said to us that in this mimetic of the human lung, that there was potentially some type of paracrine interaction. We did the same thing in the Bleomycin model, which is enriched for these macrophage populations, and we found that there was an increase in fibroblast accumulation. The fibroblasts were proliferative. The fibroblasts were immortal, but interestingly, the macrophages didn't do anything in terms of modulating the effector phenotype or the alpha smooth muscle actin expressing of the fibroblasts, saying, at least to me, that the main role of the macrophages in this system is maybe to assist with accumulation, but not actually to participate in the specification of the fibroblast cell fate. So then when you go into the Bleomycin mouse model and you do something like remove or this is a TGF-beta overexpression, but you see the same thing in Bleomycin. If you remove the macrophages or change the phenotype, there's a reduction in collagen accumulation. Similarly, if you increase the macrophages by knocking out a gene that regulates macrophage, sort of keeping them still or keeping them not activated, you see that there's an increase in the fibrosis, suggesting that both loss of macrophage homeostasis or gain of too much function could be contributing to fibrosis in the mammalian lung. So I guess the model that I'd like to put forth is that in the setting of what's happening in pulmonary fibrosis, so you have a macrophage, but it's not actually macrophage-centric. What's actually happening is, so here's the alveolus, here's the fibroblasts, is you have your epithelial cells. Epithelial cell gets its lightning bolt that Andy mentioned before. Epithelial cell apoptosis, macrophage gets activated, and then macrophage activates the fibroblasts to proliferate and to accumulate. And I think that now that there are some macrophage-targeted therapies that are emerging, this will be really interesting to see if it plays out. I hope that it will. It's probably not the only thing that's going on, but it's very encouraging so far. So in conclusion, just I think take-home points for you all today. Cells of the adaptive immune system, I don't think that they're the whole story, and right now it's difficult to see how we could modulate them. Cells of the innate immune system, though, currently it's a very active area of investigation, might be critical regulators of fibrosis via their interaction with structural cells and with other cells in the lung. And I guess I didn't have a third point. So I'd like to just end with acknowledging all the people in my lab who work really hard, my collaborators, my colleagues in the ILD Center, my funding, and definitely, of course, the patients who make these studies possible, because patients come in every single visit. They know we're going to ask for blood. Their spouses offer their own blood as healthy controls. It's really, really wonderful, the patient engagement that we have. So I'd really like to give the patients a hand, because that's really what this is all about. And that way I guaranteed a lot of clapping for me too. So thank you.